Hello, this is Pat Riley.
And this is Andrew Lavak. And this is photosynthesis. Yeah.
The purpose of this experiment is to observe the reaction that takes place within the purpose of this experiment is to observe the photosynthesis reaction from the Alodia plant.
This is Data Studio, and we use that to see the rates of photosynthesis and temperature. This is a GLX, which measures the dissolved oxygen and temperature. This is a temperature probe, dissolved oxygen probe. And that is the Alodia plant inside of water. And that's a really strong lamp. Photosynthesis is when carbon dioxide, water, and sunlight all enter a plant, and then the plant undergoes dark cycles and light cycles and produces oxygen for us to breathe and glucose that the plant uses as food and energy.
We predict that the plants will produce more oxygen through the photosynthesis reaction.
Here are our results, shown in blue. As you can see, the oxygen concentration actually dropped over the 15 minutes. We're not exactly sure why this happened, but we can assume that maybe it had something to do with the plants being in water and the oxygen molecules possibly bonded to some other substance in the water, but we are not certain.
It took oxygen out of the water.
Here again is our graph. We learned that Alodia gives off oxygen in little bubbles. These bubbles could have possibly been absorbed by the wrapping on the Alodia, or they could have popped, or maybe they floated to the top and were not registered by the dissolved oxygen recorder located at the bottom of the little cup. This is one possible explanation to why it did not work.
This lab failed.
The reason why our graph showed a decrease in oxygen concentration is because while the plant was photosynthesizing, it was also respiring. That means it was taking in oxygen faster than it could put out oxygen through photosynthesis. Boom!
We could get better results by using a plant that would not respire.
Photosynthesis is very important to us because the plants photosynthesizing give us oxygen, and
which we respire.
No, we use oxygen to breathe, and the plants also make their own glucose so the plants can stay alive, and then we eat plants. Well, actually, we can eat the plants, or animals eat the plants, and we eat animals. So plants are basically the beginning of all they allow us to stay alive, and that is only possible through photosynthesis.
I'm Daniel Perea-Barchi.
And I'm Dris Casus. And this is our lab on photosynthesis. We'll see you later.
This is our photosynthesis lab. Right now we've got one of these GLX devices hooked up to a thermal imaging thing and an oxygen reader and whatever temperature thermometer. And it's projecting data right now onto Data Studio. The top one is, I think the top one is dissolved oxygen. Yeah, dissolved oxygen. The bottom one is temperature. So right now there's no plant in there. So this is a control. So the lines should be about even, which is what they are. And yeah.
Our project, we have a
Photosynthesis is when the plants take in sunlight, CO2, and water, and they produce oxygen and glucose, which we are trying to replicate in the Alodia plant, as you can see.
For our control, the oxygen and temperature levels remain about constant. So for
the temperature increased because of the heat lamp that we had shining on our experiment.
But we predict with the plants in the little tub of water that the oxygen level is going to rise because as the plant takes in energy from the light, it creates oxygen. So in our first control right there, the green one and the red one, we only did it for 5 minutes and the temperature and dissolved oxygen level remained fairly constant. The temperature was slowly rising, but that was because of this heat lamp here. So in the second one, where we had the plant in the tub of water, the temperature rose as well at seemingly the same rate as it did before. And, I mean, the oxygen level looks like it remained fairly constant, just really jagged. So yeah, same results.
Our results show that the O2 level when we had the Elodia plant seems to remain pretty constant. We think that this is because the plant has reached a balance between the amount of oxygen being absorbed and the amount of oxygen it produces. That's why it fluctuates, but it still seems to remain the same in the basic average oxygen level inside the water.
Another experiment that could be used to get better results or better demonstrate the process of photosynthesis would be to get a land-based plant instead of a water-based plant, put it inside some sort of isolated box and measure the oxygen and temperature readings in there. Because maybe with the water and the plant, it develops a sort of, I don't know, equilibrium with its surrounding environment, so we could see if it does the same for a land-based plant. Our hypothesis may have been wrong, for we judged that the oxygen levels with the plant in it was going to rise. But we learned a lot from this experiment. Over to you.
We learned that from the data we collected, we can tell that the oxygen did not rise, and maybe the plant reached an equilibrium, which is very interesting and new to us because we did not expect that. And I think we learned a lot about photosynthesis, and especially photosynthesis and dissolved oxygen in water.
Alright, my name's Robert.
I'm Steven.
And I'm Tim. And we are Loyola APES class doing photosynthesis tests.
Alright, so what we're doing here today is we're trying to indicate the factors that affect oxygen production in photosynthesis. So in other words, we're trying to see if anything in a certain environment affects the oxygen levels. So for example, we have right here, we have a container filled with water, and inside we have the environment. First, we're going to test it and see how the levels are before we add the plant and see if that changes the oxygen levels. So we have right here, this is our temperature probe to see if the temperatures change with the plant, which we are going to add later. And this is the oxygen level probe, which checks the oxygen levels. So later on, if we see changes, we will know with this probe.
Alright, photosynthesis is a process in which several reactions where carbon dioxide and H2O with sunlight will produce glucose and oxygen. The sun provides energy for most living organisms, but few organisms can capture energy and transfer it to other organisms, such as this aquatic plant. Animals can warm themselves in the sun, but the energy is not incorporated in their bodies. Only plants, through their chloroplasts, can absorb the energy from the sun, incorporate it into molecules, and store it for other organisms. This process is known as photosynthesis.
I predict that the plant will cause the dissolved oxygen levels to rise slowly while the temperature remains the same. This will happen because the probe will pick up the oxygen released by the plant into the water. The temperature will stay the same because we are still putting in the same amount of heat into the system. Here are the results of our experiment. As you can see, the dissolved oxygen slowly went up at a similar rate to the baseline testing when we didn't have the Elodia in. And then, when we look at our temperature, the same thing also occurs. The rate of temperature increase is equal to the rate of temperature increase without the allodia.
Okay, so as you saw in the previous graph, the levels were parallel, but at the same time they were both increasing. Now the reason they were both increasing was that when we started the experiment, it already had bits of allodia, so they're both going to be increasing, but since we added more, we started at like a higher level, so that's why they ended up being parallel. Now the reason that they both, you can just...
Another experiment we could use to get better results would be for water. At the beginning, we could start with clean water instead of water with already some allodia inside. And secondly, we could have a different temperature of water that to begin with, whether it be cold, warm, or hot, so that we could determine different oxidation levels.
2, 1.
Oh, well, learning about photosynthesis was important because we saw how difficult it was actually to test and also how meaningful it is in everyday life. For example, if photosynthesis did not occur, ecosystems such as, I don't know, a city,
swamps,
puddles,
fish can't breathe
forests can't survive because you need the inflow of oxygen from the plants and humans and other organisms to do the same in reverse.